Genes are DNA. Now you have heard this line many times, but what does it actually mean? Now before we jump into genes and genome, we need to build the foundation. And today in this video, I will explain what is DNA, what is a base, a nucleotide, nucleic acid, gene, and finally how the human genome is actually organized. And by the end of this video, everything will connect like a proper molecular story. Let's start from the smallest level, DNA. Now every plant, every animal, every human, even the bacteria surrounding you, they are built from the same four letters: adenine, thymine, guanine, and cytosine. And from these four letters, nature writes the entire story of life. Today we begin molecular biology from its foundation, DNA. DNA stands for deoxyribonucleic acid. But right now, forget the long name for a second. Remember, DNA is information, not flesh, not protein, not energy, but information. Just like your laptop stores data in binary 0 and 1, cells store biological instructions in DNA. DNA is made up of repeating units called nucleotides, and each nucleotide has three parts: sugar, phosphate, and nitrogenous base. So always remember this workflow. From base you form nucleoside, from nucleoside you form nucleotide, and from nucleotide you form nucleic acid. Now let's start with the smallest unit, that is base. Now imagine building a house. Obviously you don't start with the house itself. You start with bricks and then you just carry on. In mol bio, the smallest brick is the nitrogenous base. A base is just a nitrogen containing structure. That's it. In DNA, we have four bases: adenine, guanine, cytosine, and thymine. Now, this adenine and guanine are double ring structures and they are called purine, whereas cytosine and thymine are single ring structures and they are called pyrimidine. Remember these important points. In RNA, instead of thymine, uracil is present. Now, another very important point to remember here is that adenine always base pairs with thymine, whereas guanine always base pairs with cytosine. In adenine, there are two hydrogen bonds between A and T, and in guanine and cytosine, in between them, there are three hydrogen bonds. And that's why breaking this three hydrogen bond of G and C is way harder than breaking the two hydrogen bond of A and T. Remember this important point. What is a nucleoside? Now imagine you attach the base to a sugar and you form nucleoside. In DNA, the sugar is deoxyribose, whereas in RNA, the sugar is ribose. So when you combine adenine and ribose, you form adenosine, and when you combine adenine and deoxyribose, you form deoxyadenosine. Now why it's called deoxyadenosine, I will say after some time. But right now, let's start with the next one, that is nucleotide. Okay. Now here we add the final component: base, sugar, and phosphate. Now the moment you add phosphate, you form nucleotide, and when only base and sugar is there, that is called nucleoside. So base and sugar, nucleoside, base, sugar, and phosphate, you call it nucleotide. And this is the real building block of DNA and RNA. So adenine, deoxyribose, and phosphate, you form deoxyadenosine monophosphate, which is a DNA nucleotide. Now, a nucleic acid is a long polymer made up of many nucleotides joined together. Now, DNA, as you can see here, the DNA nucleotide here, there is one nucleotide and there is another nucleotide, and they are connected here by the phosphodiester bond. It is the first carbon, second carbon, and this is the third carbon. Now here you can see that this bond is formed between the 3' OH group of one nucleotide and 5' phosphate of another nucleotide, and this creates sugar phosphate backbone, and that's why DNA synthesis always occurs in the 5' to 3' direction. Now why it's called acid? Because of this phosphate group, which gives acidic properties. Now there are two major nucleic acids, DNA and RNA. Now why it's called deoxyribose nucleic acid? Now if you notice in the 2' carbon, there is only H group is present, no OH group, only H group. As a result, this is called deoxyribose sugar. Now, if we add one O here, one extra O at that 2' position, this extra O will make it ribose sugar and we will call it RNA. Now DNA lacks this 2' OH group and that's why it is called deoxyribose sugar. Now one thing to keep in mind is since RNA has this OH group at its 2' position, that's why it is more reactive and less stable. And since DNA lacks this 2' OH group, so it's much more stable and extremely good for long-term storage. Now what is a gene? A gene is a sequence of DNA that produces another nucleic acid, RNA. In protein coding genes, that RNA in turn codes for protein. So this flow of information from DNA to RNA to protein is called central dogma. Remember, this flow of information cannot be reversed. Now, a gene mainly consists of two things, exon and intron. Now, exon sequences are obviously conserved. By definition, a gene starts and ends with exon, and introns, as you can see here, are the intervening sequences that is present in between the exons. Now, in the mature protein, the introns are actually absent, and they are removed during splicing. And exons are the conserved sequences who are actually responsible for making proteins. And this is the reason why introns can evolve rapidly than exons because of the lack of selective pressure to produce proteins. Now what is a genome? Let's zoom out. A genome is the complete set of DNA in an organism. If gene is one chapter, then genome is the entire book. In human, there are around 20,000 protein coding genes. But do you know what is the most shocking part? Only a teeny fraction actually codes for protein. Only 1%, as you can see here, they codes for protein. The gene represents around only 25% of a genome, and only 1% they codes for protein. And the remaining transposon, other intergenic DNA, they constitute the most. And by repetitive DNA, I mean the sequences which are present in more than one copy number in the genome.